good morning, good afternoon, good evening, everyone. And thank you, Novakim. Thank you, Dr. Ravi and Mr. Punevanam for the kind introduction. And it's a privilege to present this in front of this august audience about our research work. Today we are going to discuss about the use of bacteriophages, which is a safe, natural alternative to antibiotic to control the pathogenic Vibrios in aquaculture. So now, as Dr. Ravi was discussing, Vibrio is a very notorious pathogen in aquaculture. So there are several problems, and many of them, I think all of us are aware. One is luminescent vibriosis, and second is EMS or AHPND, which is a major problem in industries in Southeast Asia and also in Latin America. And the third problem, what we have, which is rampant in India, is white fecal disease, where Vibrio is again one of the causative agent, and running mortality, tail necrosis, and loose shell syndrome, swollen hindgut. These are all some of the problems Vibrio is causing. Today, if you take up the diseases, bacterial diseases, I would say that 70 to 80% of the problems is mainly because of the Vibrios. Now, this is a picture which depicts the luminescent vibriosis, where the vibrio itself starts glowing. Some of the vibrios, like Vibrio harvae and campylei, they are fluorescent. So the animal starts glowing in the presence of this vibrio, but not all the vibrios are fluorescing. Some of the vibrio are non-fluorescents as well. And the problem which is devastating today is white fecal disease. So it starts with the floating of fecal matter on the water surface. Then the animal starts, it reduces the feed intake. Hence, you see size variation as you see here. Then you can also see the white fecal matter in the gut. And then we also see the loose shell. This is one of the later symptoms. We see loose shells, and then there is low mortality. The problem here is that the animal starts, there is no sudden mortality, hence the farmer does not harvest. The animal keeps taking the feed, hence, there is a continuous loss to the farmer also in terms of his running cost and the feed cost. So, which is a major problem today. And coming to white fecal and the causes of white fecal disease, white fecal is not caused by a single causative agent. White fecal is a combination of three factors, as reported by Dr. Fernando from University of Arizona. This was the research done in the Indian scenario, where he says that white fecal is a combination of EHP, which is a protozoan, and the second factor is Vibrio, and the third factor is the stress. The stress could be in the form of variation in temperature, high stocking density, high organic matter. All these can lead to the development of white fecal disease. So, white fecal is a combination of three factors, and the one major factor being Vibrio. Hence, if you could control Vibrio and if you could reduce the stress on the animal, EHP also could be managed better. So, this was the paper published in the year 2021, where under Indian scenario, the estimated loss due to EHP was around $567 million in India alone. So, which is huge because of the EHP. Now, the next disease, which is prevalent in Asian countries, is running mortality syndrome, which is also caused by Vibrio. So, it starts, the stage one is the antenna is red and necrotic, followed by red tail and red legs. Then the animal turns yellow, the cephalothorax becomes yellow. Then it turns to reddish yellow cephalothorax, then anorexia, that means the animal stops feeding. Then we have mortality. So the mortality goes on continuously day after day. And Vibriosis has not left the hatchery production also in peace. So it's a highly notorious pathogen. In hatcheries, there is a setback in the seed production because of the Vibrios, and there is a big economic loss in the hatcheries as well. And the chronic mortality because of vibriosis can go up to 30% under stressful condition, even in hatchery conditions. So it's a pathogen which is universal to all stages of shrimp life cycle, starting from nauplife larvae, post-larvae, everything. And even the adult, all stages of shrimp are affected by Vibrio. So why is that Vibrio is the major problem, but not the other bacteria in the aquaculture? Number one, Vibrio is halophilic. That means Vibrios love salt. Many of the Vibrios can grow even at salinity of around 10% and above. So Vibrios are able to grow even in the presence of high salt. So when we say salt, supposing salt is used as a preservative in many of the preparations. So that means when you add salt, it arrests the growth of many bacteria. Whereas Vibrio is the one which loves salt and it happily grows in the presence of salt. Hence, and also we do the culture with marine conditions or with high salt conditions. Hence, Vibrio is the major pathogen. Vibrio is responsible for more than 80% of the bacterial diseases in aquaculture. Second is the temperature, what we use in our country, or the temperature of our country, temperate countries, which is around 31 to 32 in water, is highly favorable for the growth of Vibrio. And third, there are several inputs, both in the hatchery and also in the farm, which is responsible for the growth of Vibrio. So now let me take up the hatchery. In hatchery, we add Artemia biomass, and we have a lot of eggs which are not fertilized, dead eggs and eggshell. These are all the ones which are building to the organic matter, and these are a beautiful medium for the growth of Vibrio. And again, we have several moltings which is happening in the hatchery. 5 stages are there. There are 5 molds. So if you have stocked 2 million nopli, and there will be 2 million into 5, 10 million exuvia, which is the chitin. And the chitin, which is the mold, which is coming from the animal, is a very good medium for Vibrio. Most of the Vibrios are chitinoclastic. That means they can happily grow in the presence of chitin and then multiply well. So hence, these molds contribute to the growth of Vibrio more than any other bacteria. And again, it is not left algae. Even from the algae tank, you can see that in the right, there is a plate which shows the growth of Vibrio. So these are the algae which is plated onto TCPS agar, where you see growth. And also the fecal matter, dead larvae. Supposing we stock 2 million nopli and the final survival is only 40%, there are 1.2 million dead larvae there in the tank. So these are also responsible for favoring the growth of Vibrio. And again, the sediment in the tank bottom, dead Artemia nopli, is also responsible for the growth of Vibrio and Artemia shells. So, these are the factors, or these are the organic matter which contributes to the growth of Vibrio. And then what are the symptoms? Here we can see on the left side, we have a animal where you can see the necrosis. And this is the closer version where we can see in the PL, there is a necrosis of the, where we can see the necrosis of the legs of the animal. So here also you can see, on the top, there is a slow atrophy of, this is hepatopancreas of 3 animals. You can see that, it is atrophying because of the presence of Vibrio. On the right also, you see almost negligible hepatopancreas, which is seen in the animal. Also, as I told you, Vibrio has not left the eggs as well. So we can see that, under electron microscope, the eggs are loaded with Vibrio. So it is there in all the stages. It is there from the eggs. The good source of Vibrio into the tank is our nopli. So nopli is loaded with, your water may be clean, your tank may be clean, but nopli comes with loads of Vibrio. And again, Vibriosis in the grow out. So in the grow out culture, we have, again, several sources through which Vibrio can come into the pond. Number one is the feed, uneaten feed, the vitamins, additive, all these build up the organic matter, and this is responsible for the growth of Vibrio. And the second is we have, as in case of hatchery, we have thousands of post larvae which has been, stocked. Their feces, their metabolites, their exoskeletons, their molds, and the dead post larvae or the dead juveniles also favor the growth of Vibrio. And again, other biofilms and zooplanktons and all those in the pond are also responsible for the buildup of organic matter. Hence, the pond is like a bank. The shrimp pond is like a bank where there is only deposit, there is no withdrawal. So unless, the proper management practices are followed, the organic matter buildup favors the growth of Vibrio. So now, why are we talking about phage therapy in aquaculture? So the first part of my talk was the Vibrio and the problem caused by Vibrio and how does it become the primary problem in the aquaculture. Now, why is that we are considering it phage therapy in aquaculture? Number one, currently antibiotics have been used to control many of the bacterial problems. Forget Vibrios, even in human or even cattle or poultry, everywhere, antibiotics have been extensively used for controlling the bacterial issues. Now, the main issue with the antibiotics is because of the overuse and abuse of antibiotics. Now, there are a lot of multiple drug-resistant bacteria which is there in the circulation, which in turn enters into the human system. And today, our export authorities are very vigilant, and the residues of antibiotics are leading to export rejection. And farmers are very much aware, today they cannot use antibiotics to control any problem in aquaculture. And also, the companies which were working on antibiotic development henceforth, all these years, there was a magic bullet. It was like, the earlier it was not, the moment it is discovered, it was a magic bullet. It was curing many problems. Hence, companies really made millions of dollars or billions of dollars. But today, any antibiotic which comes to the market gets obsolete. It becomes resistant within 1 or 2 years. The companies are not able to recover the money what they have put in development. Hence, newer antibiotics are not coming and the pipeline is drying. Antibiotic pipeline is drying. So the next problem, this is what I was telling you, AMR spread. That means, by misusing the antibiotics in aquaculture, animal husbandry and poultry, and also in human, the resistant strains, and also in agriculture, resistant strains go between these species. If you're using it in shrimp and there is a Vibrio or there is any other bacteria which is having multiple drug resistant gene, it gets transferred to the people who are handling it and who are consuming it and so on. And later, as we go by, these people are not going to respond to any antibiotic what we are going to use whatsoever. So hence, this is going to be the major problem. If you see, this is the projection which has come in the review of antimicrobial resistant. So by the year 2050, the death due to antibiotic resistance is going to be 10 million, which is going to be the major killer in the world than cancer. So today it is around 700,000. This is going to become 10 million by 2050. So we are in a dire situation where the antibiotic misuse has led to the major problem in the world today. So people are aware and the antibiotic use is getting curtailed day by day. Now, what are the issues which we have in aquaculture? We have bacterial disease. We have losses because of bacterial diseases. And many of these strains or Vibrios are resistant to either one antibiotic, and some of the Vibrios, what we are handling, are resistant to more than 5 antibiotics. Hence, companies and the countries are well aware of the problem what we are having. Hence, any residual antibiotic is leading to export rejection. So these are the public health issues which we have because of the use of antibiotic. And again, there are no new antibiotics which are developed. And the other problem is shrimp by itself has very poorly developed immune system. So you cannot vaccinate these animals. They don't have secondary memory to take up any vaccine. And also it's not possible to vaccinate these millions of animals in the tank. So, vaccination is ruled out. Antibiotic is ruled out. Probiotic, yes, it has, but it is not a very specific, mode of action. So do we have an answer to tackle these problems? Do we have something? Is Phage our answer? Is what we are going to see in the next slides. So what is a bacteriophage? By definition, Bacteriophage or obligate intracellular parasite that multiply only inside a bacteria by making use of some or most of the host biosynthetic machinery. So the phages use the nutrients, they can use the nutrients only from within the bacteria. They cannot grow freely in any environment. So a phage typically carries a head, a body, and a tail. And these tail has got some tips or some fibers which can bind only to the bacteria it is intended to bind. That means it's like a lock and key. The tail of the tail of the phage has a key or a structure which can bind to a specific bacteria only. So, bacteriophage intended to kill Vibrio will kill only Vibrio, will not kill any other beneficial bacteria. So, it's a very specific target, or we call it a surgical strike. It cannot kill anything other than the intended bacteria. And these are the most common and diverse entities in the biosphere. Phages are distributed wherever there is bacteria. So you see a very dirty sewage where there is a lot of bacteria, you also have lots of phages there. So it is the nature's control of bacteria. It's naturally present to control bacteria in nature. So it is estimated that, if you have one Vibrio harvey somewhere, naturally there are 10 phages which is present for that. So it is so prevalent in nature, and what we are trying to do is to select the best phages and give it as a treatment to the. Where do you find these phages? As I said, phages are everywhere. It is there in your intestine, it is there in running water, it is there in sewage, it is there in the soil. To an extent that, it has around 10 to the power of 9 per gram of particles of soil. It's not for one bacteria, it's a mixture of phages for several bacteria. So phages are there everywhere. And what is phage therapy? We use the cocktail or mixture of the best phages against a particular bacterium to eliminate the bacteria in a natural way. So this is what is phage therapy. This therapy is not new. It's an old therapy. The first evidence is reported in the year 1896 by a scientist called Hankin. So he said that, people who are he reported in his paper that people who are drinking water from Ganges or people who are living near Ganges were not affected by cholera epidemic, which was present everywhere. Elsewhere, people who are away from Ganges were affected by cholera, whereas people who are drinking water from Ganges or people using water from Ganges were not affected by cholera. So he felt that there is something in this water which is protecting them. So he took the water, he filtered it, sterile filtered it, and and he also heat inactivated. So he said there is something in the water which goes through sterile filter, but it is getting inactivated by heat. That means it is a living entity which is protecting these people against cholera. Although he did not say it is bacteriophages, it was the first reported evidence of phage therapy in the history. Now, what had happened was after the early 1900s, when the antibiotic era started, this therapy died out. But even today, countries like Georgia and Russia are using phage therapy for control of even human diseases. There are hospitals for phage therapy to control the human diseases as well. So this therapy is not new, but today, when the antibiotic popularity is going down because of the issues, phage therapy is getting relooked. So now, how does a phage kill a bacteria? So if you see here, this is a bacteriophage. Now, this sits on a bacteria. We consider this could be a Vibrio. A phage sits with its tail fibers onto a bacteria. Then it injects the DNA, and then the phage, you can see that the DNA goes inside, and the phage multiplies inside a bacteria using bacterial machinery. And due to some protein which is released by the phage and due to the pressure buildup, the bacteria bursts and releases the phage. So it kills the bacteria from within. So it is called bactericidal action. This cycle continues till the bacteria is eliminated from the system. So I have a pictorial representation. So we can see that the blue one is the bacteria And this yellow one, which is sitting here, is the phage with the tail fibers attached. The next stage, they inject the DNA into the bacteria. In the third stage, we can see that the phages multiply inside the bacteria. The bacteria swells because of the pressure, and then it kills the bacteria from within. So I have a short video. I hope it plays. I don't know, there is some. Let me just try to play a video.
Phage finds its victim. It connects its tail fibers with receptors and uses a sort of syringe to puncture the surface. In a weird motion, the phage squeezes its tail and injects its genetic information. Within minutes, the bacteria is taken over. It's now forced to manufacture all the parts of new phages. They only stop when the bacteria is filled up with brand new phages. In the final step, they produce endolysin, a powerful enzyme that punches a hole in the bacteria. The pressure is so high that the bacteria sort of vomits out all of its insides and dies. New phages are. Each finds its victim. It connects its tail fibers with receptors and uses a sort of syringe to puncture the surface. In a weird motion, the phage squeezes its tail and injects its genetic information. Within minutes, the bacteria is taken over. It's now forced to manufacture all the parts of new phages.
Okay.
They only stop when the bacteria is.
Okay. I think there is some problem with the video. Anyway,
audio is there, but
video is not there. Anyway, it is the same thing which I wanted to, say about the bacteriophages, the way it worked. So let me.
Phage finds its victim. It connects its tail fibers with receptors and uses a sort of syringe to puncture the surface. In a weird motion, the phage squeezes its tail and injects its genetic information. Within minutes, the bacteria is taken over. It's now forced to manufacture all the parts of new phages. They only stop when the bacteria is filled up with brand new phages. In the final step, they produce endolysin, a powerful enzyme that punches a hole in the bacteria. The pressure is so high that the bacteria sort of vomits out all of its insides and dies. New phages are released and begin the cycle anew, using the.
So that is about the bacteriophage, the mechanism by which phages are able to kill the bacteria. The advantage of this therapy is it is self-replicating. That means as long as there is bacteria, the phages keep replicating, and it is self-limiting. As I told you, phages can thrive only in the presence of bacteria. When the bacteria is away or when the bacteria dies, slowly the phage cannot live on its own because of the lack of nutrients. They also clear. And it's a green natural alternative to antibiotic. And there is no damage to the beneficial flora. So you may be having probiotics there, you may be having beneficial bacteria, which is naturally present in the gut. Phages, when you use an antibiotic, you will be killing all of those. After a treatment of antibiotics, you are prescribed with probiotics to replenish your bacteria in the gut. Whereas when you use a phage, it will target and kill only the Vibrio or the bacteria what you are intending to kill. It does not damage your beneficial bacteria. And again, phages are already numerous and ubiquitous. There is a Nature article which says the number of phages on Earth's surface is 10 to the power of 31. The number of bacteria on Earth's surface is 10 to the power of 30. That means this is the most number or the numerous living entity present on the Earth's surface. So it is already present in nature. And they are ubiquitous in the sense we have phages in surface water, we have in animal sera. Healthy humans, 11% of the human feces contain E. coli phages, 68% of the human feces contains Bacteroides flagellis phages. So that means phages are not new, it's present everywhere. We are just giving the best ones to the animal. So we are already living in the sea of phages. And then when we compare the various additives which are used in aquaculture, so this is the paper which is published in the year 2019, which are probiotics, essential oil, vaccine, organic acid, antibiotics, and bacteriophages in the different areas like application scope, amount of solution available in the market, treatment cost, weak points, reliability, ease of implementation, and amount of factors. Phages score higher in all the beneficial aspects here. So, although number of products are not there, but they form a complete circle. So, these are a better solution than any other solution which is available in the market as per this paper. And also another important thing is many of these bacteria, what we are dealing with, can form biofilms, and phages are reported to kill or disintegrate biofilm also, and then enable the killing of the bacteria. So it prevents the attachment, it prevents the colonization, it prevents the growth of biofilm, and then it prevents the propagation. So phages can penetrate the biofilm, which many of the antibiotics will not be able to do, and then enhance the killing of the bacteria. And what are the advantages of using bacteriophage therapy in aquaculture? Number one is we are using a purely natural solution or natural alternative to antibiotic. And this is the first time that we have a solution against Vibrios in aquaculture in the world. And even antibiotic resistant bacteria can be killed. And if you improve the hepatopancreas and the gut health, the yield and the mortality, yield improves and mortality reduces. And this is one strategy where you have a cidal action. Cidal means kill from within without destroying the beneficial bacteria, which none of the therapies can offer. So these are some of the research which we have done for the last 6 years. So these, you can see that we have a bacteria which is growing as a translucent lawn on which the transparent tiny dots are the plaques created by the phages. Phages mean bacteriophage means bacteria eater. So you can see that the phage has eaten the bacteria and produced a clear surface there. And phages are also, we collect samples from different parts of the world and also different parts of our country. And we see here there is a transparent lawn of bacteria on which we spot our phages. 10 microliter of phages are spotted. And we can see here, we have collected samples from different parts of the world. And phages, if you have a broad host in phages, which can kill many of the bacteria from different parts of the world. So thus we select phages which are having a broad host range and which can kill several species of Vibrio. So that is the major part of the work to select the best phages. And the phages are characterized by several means. This is one scientific. See, I'm not showing many of the scientific data. Here, what we say is we take 10 tubes. Into one tube, we take only the Vibrio. In other tubes, we add Vibrio along with the phages. We can see that the tube in which we have added Vibrio alone, it keeps growing. Whereas when we added the phages, the growth is arrested. So these are some of the studies in vitro to prove that the phages are able to control the growth of Vibrio. And we also do a lot of molecular analysis to see that there is no batch-to-batch variation of the phage behavior. And we have several phages to confirm that the phages are molecularly similar and there is no variation. So batch-to-batch, these studies are done. And again, the protein analysis is done to see that these phages, what we had is totally different. They are not the same phages. And today we have a lot of tools. We sequence the entire genome of the phages. So to understand that there is no antimicrobial element, there is no virulence gene, there is no lysogenic, we have to handle only phages which are lytic, which will not integrate into the genome. So all these are analyzed using a lot of tools. Today we are available and we sequence these phages, then we subject the phages to a lot of stability studies like temperature, chloroform, iodine, whatever disinfectants which are used. We check the stability and then we go for limited trials in the lab. So after several trials, we set up external trials. So this is one of the challenge studies, because we cannot do challenge studies in the pond, we do it in the lab. So here we take 6 tanks. The first tank, there is no Vibrio, there is no Phage added. In the second tank, we have infected the phages with Vibrio. We have infected the post larvae with Vibrio. In the subsequent 4 tanks, 3 of them contain Vibrio with bacteriophages and 1 tank contains Vibrio with antibiotics. So we can see that after 72 hours, the death in the first tank, that is the tank in which there is only bacteria, is, a lot. I mean, only 45% survived. Whereas the tank in which we have added phages with bacteria, there is almost 89 to 90% survival, which is as good as the control. So this proves, this was the first trial which we have done in an external agency where we felt that, yes, there is a good amount of protection by the phages. Then also we can see, we have done several commercial trials. This is the result from one of the hatchery, where we see that Vibrios are grown in a plate called TCPS agar, which is a selective agar for Vibrio. So on the day 1, we see that there is lots of Vibrio colonies. And as we do the treatment, we can see that the Vibrio levels are gradually reducing. And by day 3 to 4, we see that there is very minimal Vibrio to cause any infection here. So this also proves theoretically that, we have controlled the Vibrio in the hatchery. And this is one of the big trial which we have done in Papatla, where we have taken 14 ponds, out of which 4 ponds served as the test ponds and the rest 10 are the control ponds, where they have used the best of probiotics. And the 4 ponds were treated with elixir. Elixir is the bacteriophage-based product. So now, in these 4 ponds are, again, it's a biased selection because these 4 ponds always had problem of white fecal in the previous crop. So these 4 ponds were selected for the trial of elixir. The reason being the economy of the farmer, that he wanted to try in the deceased ponds. So here we could see that wherever we have used elixir, the average survival increased by 11%. FCR was better by almost 0.24. That means he had better FCR using elixir. And production per hectare also was much higher with elixir when compared to the control, almost around 1,200 kg. Here I'm not able to see the figure. In the next slide, we can see again production per lac of seed also improved with elixir. And average daily weight gain also was better with elixir, 0.16 grams in elixir treated pond when compared to the probiotic or control pond, which is only 0.12 grams. So this is the overall results. So where the survival was 87 versus 76 in the control pond, FCR was 1.6 versus 1.84. Production per lac of seed was 4,200 versus 3,700. Average daily gain was 0.16 versus 0.13. So in all the parameters, it showed that this was a better bet. And the economy, ultimately the farmer wants his money back. So the benefit to cost ratio when using elixir was 1.35. That means for every 1 rupee of money spent, he got 1.35 using elixir, and for others, the control pond, he got 1.06. So this is what is ultimately important. And this is the recent trial which is done in Nagapattinam. Trial, it's not a trial, I would say, it's a commercial use. Here what had happened was, this area had white fecal disease. We had a meeting. Just after that, there was some problems. So here, this farmer also used elixir. So here his survival went up by almost 24% when he used elixir because of white fecal disease. Again, yield per acre improved a lot, 3,900 versus 5,733 per acre kg. Then average daily gain also 0.18 in control and 0.22 in the elixir pond. And again, FCR improved drastically, 1.7 versus 1.3. And this was the trial which we have done in a nursery pond in Costa Rica last year. So where we had two ponds which were treated, one with elixir, one was not treated with elixir. And this was done in 2 seasons, one during summer and one during the monsoon. And in Costa Rica, the main problem, or in Latin America, main problem is early mortality syndrome, which is also caused by Vibrios. So here you could see that his survival improved by 8% when compared to control pond. And here in the second trial, the days were more with elixir. He could not harvest on day 30. This was the initial trial. We didn't go, we have not done the complete trial. So 45 days, here it is 30 days. Hence the survival slightly dropped. But then his weighted harvest was much higher with the elixir pond. And again, here it is 3.6 is weighted harvest when compared to 1.3. FCR also shown a drastic improvement, 0.57 versus 0.81. And beyond all that, this farmer always had EMS in his nursery. And this was the first time where the treated pond did not have EMS, whereas the control pond did have EMS. He said that, never my survival has improved or gone beyond 50%. This is the first time it has gone to 78%. And we will continue to use phage therapy as we believe we have a potential solution for EMS. So this was the comment by the farmer. So now, in a nutshell, to summarize what we have done, phages are very safe. We have understood the inadequacies. We can say phage therapy as pre-antibiotic and post-antibiotic era. We have understood the inadequacy of first era of phage therapy. Today, we have better tools, better genomic data available to understand the phages better. And we also have a lot of well-controlled animal studies done to know that phages are indeed a good solution or good alternative to antibiotic. We also have a lot of polished therapies. Poland, there is a lot of studies done to say that phages can tackle even drug-resistant bacteria, or it can, some of the studies have shown it can convert drug-resistant bacteria to drug-sensitive bacteria also. So I would say that the world is reconsidering phage therapy. It could be a standalone therapy or it could be a valuable adjunct to your probiotic. It's an ultimate clean, green, eco-friendly disease control system. So the summary of the results, many of the farmers have reported the drastic reduction in total Vibrio count, reduction in running mortality syndrome, control of white fecal disease, protection against EMS. And several hatcheries have reported the larval survival improved with bacteriophage therapy. Also, Vibrio counts have decreased in LRTs. This could be a standalone therapy or a valuable adjunct to probiotic. So hence, we firmly believe that nature itself provides the solutions for modern day challenges. Bacteriophage is the result of that belief, an effective organic solution to make food safer. This is my team and, who has worked. And again, about a bit about Aristogene. Aristogene is one of the few companies which has been recognized for its in-house R&D by Government of India, DSA. And again, I think he has already mentioned about the Innovator Award. And we also have a lot of projects sponsored by BIRAC. So we thank BIRAC for this opportunity. And we have an excellent team who works, and without them, it's not possible for us to bring out this product. So thank you, everyone. Thanks for your listening.
So thank you very much, madam. It's an excellent presentation.
Thank you.
We have a question and session now. If you have any questions, all the participants, please type in the comment box, or else you can unmute yourself and ask madam directly.
Hello, sir. Good morning. Thanks for the providing this link to participate in this. And thank you, madam. Sir, I had a doubt. Sir, am I audible to you?
Yes, Mr. Srikanth, please go ahead.
Thank you. So I had a doubt that this bacteriophage treatment you have started, madam has shown some 2 to 3 number of case studies. Actually, I could not able to remember that because it's a very quick session. So I had a doubt that It was bacteriophage treatment was started from the starting stage of seeding, or when any Vibrio colony is found in the high number. That is the doubt I had, sir. Madam, if you can answer, it will be very great.
Yeah, the first study which was done in Papatla was a prophylactic study. That means from the day 30, we started the treatment. Whereas the second study, which is Nagapatnam study, they had the problem of white fecal, which started. It's more like although we don't promote a therapeutic dosage, because these animals are really very small, their immune system is very poor. And if it is having the disease, that means when you go to treat an animal which is already with the disease, it's difficult to recover them. So we always advise them to start as a prophylaxis, or at least in the early stage. So this farmer has started after the symptoms have started. So that second study, what I've showed in Nagapatnam, is the kind of treatment study, we can say, because here there are some animals which are there in the early stage of disease. So you don't say that all the animals are at the same stage of disease. So the level of Vibrio may be slightly less in some animals. Hence, the second study was a treatment study. First study was a prophylactic study.
Okay, madam. Thank you. And another thing, maybe I was asking one or two more questions. Is it okay, madam?
Yeah, tell me, please.
The thing is, the antigen, so if you take the Vibrio, it causes the many diseases. Like one of the diseases I was knowing is white fecal syndrome. Maybe it may cause other diseases. So which part of the antigen it was triggering that disease? Why? Because this question I am asking why, because if you are killing use the Vibrio using bacteriophages, it will rupture the cells. So again, that residues will be remain in the body of the animal. And how to remove those also? Do you have any idea, madam, or can you throw some light on it?
Okay. So your question is whether the killed Vibrio can also cause any problem. Is that the question?
Yes, ma'am. Yes.
Okay. See, as we were talking, this therapy is more like a prophylaxis. Okay. So there may be an animal which is having lots of Vibrio where the phages have worked and killed the Vibrio. We may not be recovering those animals which are loaded with huge amount of Vibrio, because even if they recover, how well they perform, how their feeding is, how it is going to improve the farmer's FCR, we don't know. So we are targeting the animals which are at a low level of Vibrio load. Okay. And secondly, if you know, many of the vaccines work with the antigen model only, correct? So see, there are some bacteria where upon lysis, there may be a toxin release that can be a problem. But the protein which is released on lysis of Vibrio as such, that I don't foresee as a major problem to kill the Vibrio, because there are some proteins which are also used as immunostimulants. But we are looking at a case where the level of Vibrio is so low, less in the animal, not in an animal which is having 10 power 7, 10 power 8, which is dying. So some percentage of animals we may be losing. Correct. But there are majority of the animals which are there in the early stage of the disease. We have never seen any incidents that, these kind of problems are there. So this is, I don't know whether I've cleared your doubt. So this is what, yeah.
So if I understood correctly, it will be good to use from the starting stage onwards.
Yes.
Thank you, ma'am.
Is there any other questions?
Yes. Yes, Ravi, I'm Charan.
Yes. Good morning, Dr. Charan.
Thank you. It's all going good. My point is that the Costa Rica experiment and the result you have given, whether we are following it in the growth also. So there could be some significant difference in the growth result after using the product.
Okay.
That has to be established. Is there anything in that line going on?
Yes. They are also doing a study in the grow out, but the same farmer who has used, we have given it as a sample for nursery, but some product was left and he has used it in the grow out as well, and he got very good results, but we haven't taken because it was done, by his will. So we haven't taken the results from him, but we have completed one more study in the grow out in much more statistical way. The results are yet to come out. And again, the third study has started again in the grow out in Costa Rica.
Thank you, madam. One more point. See, this protocol where using elixir in the grow out farm or bacteriophage in the hatchery, some more clarity to the mode of the participant. As a prophylactic, we are using, what dose, how much gram per kg, how many days it's supposed to be. So some general guideline, if you are throwing some more, it will be beneficial for our audience.
Elixir we advise to use as a feed additive. This is used at the rate of 5 grams per kg of meal, 1 meal per day. And this is mixed normally with feed additives and then, sprayed onto, mixed with the feed. Maybe we can dry it for 30 minutes and then we can, broadcast. In case if the Vibrio loads are high and if the farmer is having early stages of disease, many of the farmers have used 10 grams per kg of feed, 2 meals a day for at least 10 to 15 days till the symptoms subside, then come to prophylactic. We never ask them to abruptly stop the dosage, because when you use very low dose or when you stop the dosage without the Vibrio getting cleared, you know that Vibrio multiplies within 20 minutes. Their cycle time is 20 minutes. The level of Vibrios probably overtake. If you have cleared it, halfway, it overtakes, and then with the next 1 week, the level becomes so high that again you need to use a huge dose. So never abruptly stop the treatment and give a prophylactic treatment if possible. And if there is an early stage of disease, we can give, which is 4 times the dose, 10 grams per kg of feed, 2 meals a day. And this continues for 10 to 15 days. Then you bring it to 5 grams per kg of meal, 1 meal a day. So this is the ideal way of treating.
Thank you. Thank you.
This is Chandrasekhar.
Yes, sir. Good morning.
Good morning, ma'am. Ma'am, there is any difference in low salinity area and high salinity area, how it's performing in low salinity areas? Because in Andhra Pradesh, there is low salinity culture and high salinity culture also.
Okay. See, we have tested, as I told you, when we select the phages, one of the criteria for selection of phages to check the activity under various salinity. So we have tested the phages from 0 to 70 ppt salinity. So as long as Vibrio is there, as I told you, phages cannot use anything from the water. They cannot survive or use nutrients or anything. So probably it is also not affected by the water salinity. It grows very well from 0 to 70 ppt. This is also one of the selection criteria of the phages which we have in the beginning itself. So salinity does not have any impact on the titer or the growth of phages.
Thank you very much.
Hello, madam. I had one more doubt. Like
Yes.
TSC, BSC, many countries they're checking for the TSC, BSC, like viruses, anything is there. Hope this may not have any impact on the exports.
Okay. See, as I told you, phages are, even if you don't add phages, there will be natural phages in the seawater, in whatever we are using, our hands, the handlers, everywhere there are phages. So phages is a natural component of the system. And second is, we don't call it as a virus because the virus by its whole body or the entire virus enters into the eukaryotic cell. Phages can only inject the DNA into the bacterial cells. And the phages, as I told you, cannot thrive without the presence of Vibrio. Hence, Vibrio clears from the water. The phages also clears very fast from the circulation. So, unlike an antibiotic which leaves a residue for 2 to 3 months, phages do not leave a residue. Even if it is, the levels are as good as which is not naturally present in the, hand or feces or sera of any or this one. So, since they are natural, it is everywhere. Even if you don't add phages, there may be some phages through several means.
Okay, madam. Got it. Thank you.
So there is one announcement. Just now I shared a link in the chat box. I request all of you to open that Google Sheet and enter the topic you preferred for the next webinar. It will be helpful for us to select a good speaker and good topic. So thank you. So if there are no questions, Panavaran Garu.
Yeah, Ravi garu.
Yeah, we can close the session.
Yeah. Thank you. Nice, Ravi garu. Ravi Aqua Academy and Novacom Biosciences together just within few days. I should say within 3 days, I think. Even I was slightly worried, but you said no, go ahead. And thankfully, Madam agreed because she is the main person, the speaker. So instantly she agreed, and it has gone very well, ma'am. And Madam was telling only one point I want to tell that Madam told that Phage biotic, bacteriophages, it is not new to this drug. But let me tell you, ma'am, I am in this industry for almost 30 years, and after my PG, first time I heard about bacteriophages. In my post graduation, we studied about bacteriophages. Wait here, in aqua industry, we have seen antibiotic, probiotic, prebiotic, this, that, but this Phage biotic, it is a new word for aqua industry. And we are very happy that Aristogene from Aristogene, we have developed this technology and we are also part of it. That is what Ravi Aqua Academy as well as Novacom, we deal with your product and we are very happy to, we are very grateful to you. And to sum up with this, see, like Ravi Garu was telling that Novacom and this Novacom Biosciences, it is a 1 year old company, and along with Ravi Aqua Academy, this webinar series will go ahead, it will keep continuing. And let me tell, most of the participants in this webinar, around 100 came, and very in a very short period, so we could mobilize this many. I feel that it is good. So many good number of people and from various countries they have joined. This industry, this aqua industry, is a three decade old industry almost. From my knowledge, from my understanding, I can say that the first from 1990 for Indian aqua industry, first 2 decades it was ruled by mostly by Black Tiger as well as Indigas and little bit of Scampi. Post Vanamai introduction, aqua industry has grown like anything. It has grown by leaps and bounds, robust to growth, and now the annual production in India it has reached the range of 7 or 8 lakh tons per annum. But there are serious concerns. Okay, we have seen it is always a roller coaster ride, and there were speed breakers, there were hurdles in this industry. It is not new for aqua people, but recently though productivity has increased, water study has increased, and for the past 4 or 5 years, we have seen failure rate has kept on increasing. Failure rate has kept on increasing. Average harvest size in the beginning of the Vanama, it was 40 count or 50 count, 20 or 25 grams. It has been steadily coming down now. It is believed that it is now at 12 grams or 10 grams of LBW in most of the areas in India. So impact of COVID, global economic slowdown, and other geopolitical issues or unexpected global production from like countries from Ecuador and other countries. These kind of factors have led to a serious problem for the aqua farming community. See, certain things are out of our hand. It is beyond our hand. Say like volatile farm gate prices. 1 month back, it was 100 pound. It was at 290 or 300 rupees. Now it is 190 or 200 rupees. Input cost for the farmers, it has gone up like anything. These are all out of our hands. But what farmers can do? See, like cost control, production cost control, and disease management and feed management. See, like recently, I have been visiting Kabali market for the past few years, 4 or 5 years. One of my good friend is a dealer. He sell around 2010, more than 2000 tons in summer crop. And he was telling last week when I met him, he was telling that he has sold it more than 2000 tons, and 90% of the farmers have made a successful summer crop. And his average FCR for 2000 tons of feed, average FCR is, he was telling 1.2 only. 1.2. And he says that more than 90% of the farmers have been successful. How it happened? And let me tell you, I know I visited most of the aqua farming villages in Kavali. They are all small farmers. See, there are so many reasons, but let me tell you only one point that it is the crop holiday in Kavali market. They strictly they follow this for 3 4 days. See, It is like we have to keep on interacting and we have to keep on debating. So that will be helping this aqua community, farming community. Novacom will continue to take efforts to help the farming communities. We'll take all of the effort. We'll bring in a lot of eminent personalities of aqua industry to sensitize the farming communities on various technical aspects, and we'll keep on discussing about all the burning issues. I'll not take much of time. Once again, thank you so much. Thank you, Dr.C.R.Subhashini madam. Thank you, Ravi Academy, Ravi. You have done it a superb job. So thank you, team Novacom. Yes, let us meet soon to discuss on a different topic. Thank you. Thanks for this opportunity. Thank you so much.
Thank you, everyone. Thank you.
Before ending the session, I want all of us to congratulate Madam by typing more power to you, MPTU in chat box, MPTU. Congratulations, Madam.
Thank you. Thank you very much. It means a lot. Means a lot to us.
Thank you.
Thank you. Thank you, Nirmal.
Thank you all. Bye.
Thank you.
Thank you. Thank you.